Hello and welcome to Lecture 2 of MOL213, Developmental Genetics. My name is Fergal O'Farrell and I'm here to take you through it. So if we have a look at the learning goals for today's lecture, we want to appreciate the importance of gene regulation during development and importantly the techniques used to monitor the expression of developmental genes in the time and the place. We want to expand our terminology before we go further. So we're going to learn about specification and determination, as well as the different ways of inducing cells to change cell fate, and whether they're competent to respond to those signals. So induction and competence. We're also going to start looking at the basics of the mechanisms that underlie pattern formation and some introduction to important cell signaling pathways in developmental biology. We're also going to have a look at the early development or an introduction to the early development of model organisms, and have a question mark here because it might be that this lecture gets a bit too long and I take the model organisms part later. Okay, let's get started, shall we? So just to start with a recap of the last lecture, we had some fundamental questions, and one of them was how does an egg develop into a human embryo with more than, so going from one cell type to a complex organism with more than 200 cell types and a very distinct body pattern and developed tissues and organs. And we said that they discovered that the cells were dividing and differentiating, and as well as patterning and morphogenesis was important for this. And we also came with the question then, how are the different cell types generated from a fertilized egg? And we came with the answer in the end that different cell types are generated by differences in gene expression. And then indirectly we can ask the question, well, if the zygote, the single cell, through this process is capable of generating embryos with different cell types, does every cell contain the same genetic information as the zygote? And this led to experiments then by a famous scientist called John Gurdon. Performed experiments in late 50s, early 60s on Xenopus, the frog, which would pave the way for a lot of science fiction discussions which are becoming a reality on human cloning and the potential of cloning yourself. So this is quite interesting. So let's have a look at it. So what he did was he took unfertilized egg from the frog and he destroyed the DNA in the nucleus with UV radiation. And then he took cells from the adult animals from either the skin or in the case of the tadpole, he took a cell from the epithelial cells of the intestine, which were then, they were diploid, right? Differentiated cells. And he removed the nucleus from them and he put it back into this egg in which the nucleus had been destroyed. And what he saw was that this now differentiated nucleus could give rise to a tadpole and in some cases even adult frogs. So this is a pretty remarkable experiment, mostly just physical manipulation of the nuclei and the eggs, but it's still pretty cool and groundbreaking. So what it could show was that the nuclei in the differentiated cells still contained all the genetic information required to make a complete animal. So yes, all the cells contain the same genetic information as the zygote. This gives rise to the idea of genetic equivalence. All the somatic cells in the body of a multicellular organism contain the same set of genes, right? That genes are not shuffled around between cells and gained and lost and transferred during normal development. But the activity, the transcription of these genes is different. So we have differential gene expression in the different cells. When they're differentiated, it takes a very special set of circumstances to bring them back to an undifferentiated state, which is what has happened here when they transfer the nuclei into this unfertilized egg. I've put in here instructions, and that's to indicate that the DNA contains a set of instructions how to build an animal, and those instructions are not lost when the cell is terminally differentiated, but they're not used anymore. A new gene expression program has been turned on there, and they don't need to make a new animal again. Okay, this is not something that will be on the exam, but I just want to point out, I don't know if everybody's heard about the most famous sheep in the world, which is called Dolly, which this is just taken from Wikipedia, that Dolly was the first mammal that was cloned from an adult somatic cell using the process of nuclear transfer as pioneered by John Gurdon in his frog experiments. And in the case of Dolly, she had three mothers. One provided the egg, another provided the DNA, and a third carried the cloned embryo to term. And I can't remember how many thousands of eggs they used in this experiment to get one to survive, but in the end they got it to work. So it's not so easy. The first successful cloning of a primate species, so some monkey, was done using the same method in 2018. So things are moving ahead. And okay, yeah, here I wrote down how many attempts there were. Yeah, so in 1996, Dolly was cloned first, and she was the only lamb that survived to adulthood from 227. So approximately 1 out of 300 were successful. So not too bad. By 2014, Chinese scientists have a 70 to 80% chance, so that's gone up dramatically when they were cloning pigs. And at the moment, a Korean company is producing about 500 cloned pig embryos per day. And then I just wanted to point out that it is now possible to pay to have your animals cloned. Maybe you know this already. And a famous singer, actress, Barbara Streisand, actually cloned her dead dog, and she got these two dogs. And yeah, you can read about that if you're interested. But what was interesting was the prices. So for cloning of a dog, it's about $50,000, which is about $500,000 kroner, and they cloned cats for about half the price, just in case you're interested. I should say that when I take examples like this, that I'm presenting the facts as it is, and I'm not giving any ethical considerations to whether things should be done. I'm sort of reporting what has been done, and while some of these things definitely warrant discussion surrounding the ethics, I'm not going to take an ethical discussion in the lectures. So different cell types are generated by differences in gene expression, but all cells contain the same genetic information as in the zygote. Therefore, we need complex control regions to control the expression of developmental genes. We need to be able to switch them on when it's the right time, and we need to be able to switch them off and even keep them off. In the case of terminally differentiated cells, we do not want them to reinitiate expression. This is one of the things that could lead to cancerous growth, for example, or other diseases. And it's often said that developmental genes provide a set of instructions, not a blueprint for development. So what does this mean? Well, if it was a blueprint, it would say in your DNA how tall you will be, how long your arm will be, and it does not contain that detailed information. Rather, the genes encode for proteins, as we know, and the proteins will do the job and make certain types of cells. Those cells will then interact with the surrounding cells. So it's more a set of guidelines or instructions that are provided by the DNA, not the definite. So we can't go in and manipulate one gene for height, for example. So control regions are very important for the readout of instructions, and it's transcription factors that bind to control regions, and switch the genes on and off. And developmental genes usually have complex control regions composed of cis regulatory modules. And cis means that it's on the same strand, even if it's far removed, which often can be in the case of complex control regions, that the enhancer or suppressor regions or that bind activator or repressor transcription factors, they can be some distance from the start site of the coding region of the DNA. And this distance, this gives more room to play around with how many transcription factor binding sites you can have, right? They're not at the core promoter region, which is already occupied by the normal transcription machinery, the RNA polymerase, for example, that will give rise to the generating the mRNA from the gene. These cis regulatory control regions, enhancers and suppressors, we call them, are further from the gene and can have genes, for example, other genes coding here for normal processes that will not be affected by these long range acting cis regulatory elements. The expression of the developmental gene will be determined by the combination of transcription factors that are bound. So it's not so simple as one transcription factor is turned on and we get expression of 10 genes that have that binding site for the transcription factor. At least it's not always that simple. So normally there's, as I say, 4 to 8 control regions. Some may be for the same transcription factor, but usually you have several transcription factors that can be acting on the control region to drive expression of the gene. And each module can function somewhat independently. Well, so if we take the whole region of the whole control region and tag it to a reporter gene, we can get an accurate readout of how the developmental gene would be expressed in the developing animal. But if we just take a part of it, we will just get perhaps just a part of that expression, which is maybe the expected. But what's maybe more unexpected is that we can also get a much broader expression in terms of time and space by just taking one piece. And this indicates that a suppressor element has been lost when we clipped out or cloned this piece of DNA and tagged it to a reporter construct, that we didn't take a suppressor element with it. And therefore, when the transcription factor bound, gave a broad expression throughout the whole animal. Whereas some cells in the whole animal would have expression of the suppressing transcription factor and would normally block that expression in those cells, right? So it's a complex control mechanism underlying the expression of developmental genes. And this is what you need to get expression at the different stages and locations or specific cell types. Moreover, there is a complex interdependent network of developmental genes. So they act on each other. The transcription factors will act on the genes, and the genes that are produced can act on other genes, including other developmental genes and genes encoding for developmentally important transcription factors. So here's a couple of examples. You probably know this. We can go through it. We can have positive and negative feedback. And in the case of the positive feedback, we have an activator, which can be a signal coming into the cell via a receptor leading to activation of a pathway, which we will see examples of later on. Or it can be, for the simplest case, a transcription factor that now binds to the DNA, activates expression of the gene. This gives rise to the protein here in red, and this protein can act on a neighboring gene. It can be directly a transcription factor or something that activates a transcription factor, right? This activates gene 2, which then produces its own protein. And protein 2 can be, again, a transcription factor acting directly on DNA, or can be a part of a signal cascade that activates a transcription factor to act on the DNA, or anything that acts on chromatin, for example. The point of here is that this will be a positive feedback, so protein 2 will actually act on gene 1 to encourage its transcription to persist. So even now, if you removed the activator that activated gene 1, protein 2 will maintain the expression of gene 1 and via that gene 1 maintain its own expression. So this is a positive feedback loop. On the other hand, a negative feedback loop, again, we have activator coming in, acting on gene 3 in this case, producing a purplish protein which acts on gene 4, and gene 4 then activates gene 4. Gene 4 produces a protein that is in fact a negative regulator of gene 3 and will suppress transcription. This will lead to loss of this activation pathway where gene 3 activates gene 4. So actually, gene 4 product will shut down gene 3 expression, which will shut down expression of gene 4. So it will shut itself down. If the signal here was persistent, there would be so signal, in this case it's removed, so this will happen just once. But if this signal was persistent, there's a number of scenarios that can happen. If this protein, the suppressor of transcription of gene 3, has a short half-life in the cell and is turned over, it will be degraded, right, and gone. If activator is still present, then this loop will initiate again. Okay? If it is not present, it will stay off. Now I want to show you a film. This is in mouse, and it's in the somites forming in the embryo. And it's an example of negative feedback. So hopefully I can get it to play. Okay. So what it looks like is pulsing signal moving, pulsing signal moving from the tail towards the head. So from the posterior towards the anterior. One more time. We will look at this in more detail in, I think it's lecture 6, but we can just say that there is an activator being produced in the tail that turns on the transcription of a gene, and the activator actually is diffusing from the posterior region more anteriorly, right? It's moving, and then the transcription of gene is appears to be moving, but what happens is that the activator meets cells, turns on this loop where we get a flash of GFP, and then that loop is a negative feedback loop that will turn off the GFP expression, but the signal moves further to the next cells that can also respond to it. Okay, we will go through this in more detail later. I just thought it was interesting to give an example of some negative feedback. And then it's important to point out that the gene expression, we talk a lot about transcription, is only the first step in a cascade of cellular processes that are important to development. And we mentioned this in lecture 1, so I won't labor the point, but we talked about turning on the gene. It gives rise to an mRNA or an RNA, which can be processed into an mRNA, and this is transported to cytoplasm. You need to make the protein is modified, and then maybe the protein isn't a transcription factor which acts directly on DNA, but is in a signaling cascade, or it's a ligand that will be secreted from the cell and activate events, signaling pathways in a neighboring cell. So there will be a time delay between switching on the gene and the actual event that the gene wants to happening, if that makes sense. And I want to point out that these times can differ between species, and this is one reason why development takes longer in one organism versus another. Even though these processes are in common, the time that it takes and the rate at which they occur in different animals can be different, and this contributes to differing developmental times. It's not the only thing, but it's one of the things. And I sort of covered this again. We can have another factor is the net that affects the developmental timing is gene networks. So we can have all of this that I presented in this previous figure, this whole cascade happening to produce a product that then affects the or turns on the transcription of another gene. And it also needs to be translated into a protein eventually, and that can act on another gene. So again, this you can see that there's a wide range of possible timing differences between different species for these events to occur, even if it's the same signaling pathway in the same developmental process. So this brings us to detection of gene expression during development in situ. And in situ means in place, or in our case, in the animal or in the cells. And I'm going to talk about a few techniques here, all of which are to some extent still important today, but we can take it a bit in the order in which they were used. Okay, so one way to detect the expression of gene transcription is via the mRNA that is produced. And to do this, we use a technique called in situ hybridization. And in situ hybridization is an important technique today still, especially in some organisms that have less genetic tools available. In order to do in situ hybridization, you need to make an antisense probe using DNA or RNA that will bind, that recognize the sequence of the mRNA and bind to it. And this antisense probe has a label on it that we can then detect or visualize. And in the old days, it was radioactive probes using 35S labeling. But nowadays, we have non-radioactive approaches, include deoxygenin or DIG labeling, which gives us the development of a blue color typically. But we also have now, of course, fluorescently tagged mRNA probes. This approach in the old days, perhaps more, started with tissue sections, which were either frozen or wax sections, and developed then later to looking at whole mount embryos. And I'll show these a little bit later, but I want to point out that a couple of things here, basic things. So we make antisense probes that are the exact opposite of the mRNA and recognize that and bind tightly to it. One caveat of this approach means we need to know the DNA or the DNA and mRNA sequence of the gene we're interested in to design that probe ourselves. And then, the typical thing is we have a cloned gene in a vector and we use a type of polymerase to give a reverse transcript of that, for example, and make probes that way. But we need to have that DNA sequence already in hand. And these days, where many organisms are genomically sequenced, that's not a problem. But in the old days, that was more of an issue, right? The other thing I wanted to talk about, we're talking about tissue sections and whole mount embryos that we do this labeling on. Obviously, these animals or these embryos are dead and fixed. So it's not used in live imaging. The animals are typically fixed in formaldehyde, for example. Just so you keep that in mind. There are limitations to this approach. And I don't know how many of you have had experience doing in situ hybridization. I just thought I would give a quick description of it. We generate an antisense probe, which is then incorporated with a label. We put it on the cells, tissue, or section of a tissue that is already fixed. And then we do an RNase treatment to degrade most of the signal away, except for the double-bound DNA RNA or RNA hybrids. And then we incubate with an antibody to the signal in the case of DIG, or if it was radioactive, we could just expose them to photographic film That's what they used to do in the old days, right? And then in the case of the dig labeling, we use alkaline phosphatase to generate a color, which is written as purple here, but it looks blue in my eyes. This goes through how you do the fixing of embryos embedded in something like wax or ice, and then the sections put on a slide. They get radio labeled, and then you develop them generally on a photographic plate, and you get this kind of image. And this was really discontinued in the 90s. Most of these things where you don't have to work with radioactivity, you definitely don't choose to work with radioactivity, right? But it was what was available at the time. And then this slide just indicates that, yeah, the advancement was you could do whole embryos, and then you didn't need to section them to visualize anything. You could look at the whole embryo mounted on some cover slip. So they improved the sort of penetration technique then of how you could get both antibodies and probes into the animals. And then visualization technology also has improved over time. And basically since the 90s, this is the chosen way to go. Here's some examples. Here we have a zebrafish embryo labeled with 35S with the RNA probe. We see then when we develop the picture where we have expression of the gene. And this is then the light microscope view of this thin section. You see that the gene is heavily expressed here in this area of the embryo. These are some that I made myself during my PhD, and here we have Drosophila embryos labeled with different mRNA probes. And here we have an eye disc from the developing larva, and a brain from the larva. And the cool thing with these, apart from not having to use radioactivity, and you can visualize them through the light microscope, the cool thing was you could combine them with antibody stainings, for example. And here I use a different type of coloring agent to detect the antibody present, labeling certain cells in the eye, or here certain cells in the nervous system. You can see that the gene I was interested in at the time is expressed in a nerve, and then we label cells that are attached to the nerve in these peripheral nervous system organs. And here we have some expression in muscles, and you can see the nuclei in the muscle cell. The muscle is really a syncytium, right? It has many nuclei in one cell body. So the cell walls between the cells have dissolved, and we have many nuclei within a strand of muscle fiber. So techniques have advanced, and we have probes or antibodies tagged with fluorescent dyes that we use now more often. Here we have an example from Drosophila. It's a multiplex. This is a Drosophila embryo. It's a multiplex fluorescent mRNA in situ hybridization. So detecting the expression of several different genes and labeling them with different probes tagged with different colors. It is pretty cool that you can see the expression of genes relative to each other. This is just a similar example of 3D projection through the embryo. So that was with detecting expression of genes with in situ hybridization, a very common technique, and more people have been using antibodies and fluorescence, and I'm sure you've used antibodies and don't need much introduction to this. Here's an example from the book where they took some tissue and they serial sectioned, and then they applied the antibodies separately and colored it artificially in a computer. But you can use, there are several colors available for fluorescent detection on most microscopes, and so you can do several colors at the same time. Maybe not 5 like this, right? But you can certainly do 3 colors, 4 colors without much difficulty. And here's some developing Drosophila embryos and a zebrafish embryo where we have used antibodies and then secondary antibodies with fluorescent tags to detect the expression of proteins. So this is, of course, the product of gene transcription, right? And shows us where the protein is produced and at what time during development. Again, I can mention that like with in situ, these animals are, of course, dead and fixed using formaldehyde usually. to fix them, and then we can detect if the protein survives the fixation process, we can detect it with the antibody. So there's limitations with this approach, but of course it's extremely useful to study development. And the typical thing, for example, with the Drosophila egg would be that you collect it at different time points and perform the staining then on those. So you can either, so every hour or every 2 hours you do embryo collections, and then you can pool them and use the same antibody, or keep the time points separately and use the same antibody. And yeah, but actually over time, looking at these embryos, you learn how old they are by the morphological changes to the animal. Here you see the central nervous system is very developed, a ventral nerve cord, and the intestines are very convoluted. So this is a very late staged embryo. Whereas here is quite early, even though the markers here, I'm not sure they're the same. It might be. But you can see that sort of germ band extension has just happened. This is an early embryo. In the end, this part of it's going through gastrulation, and this part will retract and pull back here, so it will look more like this. This is basically the nerve cord will pull all the way back here, and we'll have a look at this later on. But I wanted to say a few words about, so I've been pointing out all the time that we can use these techniques in situ and antibodies, but the animals are dead. And more are coming detection of gene expression in situ in live animals. And the way to do this is by using transgenic reporters or vital dyes, and you can look at protein expression live. The vital dyes are more sort of generic kind of proteins you can detect with that, like actin, or you just label the cytosol, or mitochondrial structures are typically looked at using this. So organelles, there's some vital dyes for, but it's rare that you have proteins that there are vital dyes for. But there are ways to get at that, to express things to detect the proteins. But more common way to look at transcription is to make these reporters. And I talked about how we could chop off a regulatory sequence, and we could tag it to a reporter protein, or gene encoding for a reporter protein, for example, GFP or luciferase. And then we put that into this DNA construct into the animal, and we can see how this reporter construct is expressed depending on this enhancer element. We can then also perform tagging on genes at their locus in the animal. This here I've talked about making a construct and putting it into the animal, but we can actually use a construct that inserts the GFP into a gene of interest, and then we can see how the GFP tagged gene or protein is expressed during development live. And this is an example of that, is it? No, this is still sort of a more complex version of an exogenous construct being put into an animal. And then we can have a look at the reporter here. I think it's luciferase in the animal while it's alive. Here's just a couple more examples here. I think these are, again, this is a developing zebrafish embryo where they have tagged or they have reporter constructs with GFP in green or RFP in red to show where these genes are expressed, and in blue they've used a vital dye for DNA. So this animal is alive, and you can look at the expression real time of those genes. This is another example here. Here they're tagging the mitochondria with RFP. They look a bit purple here. And then neuro-D, some gene expressed in neurons tagged with enhanced GFP, so a GFP fluorescent probe or reporter. But like I mentioned, it is nowadays possible to do tagging at the gene locus, and CRISPR-Cas has been one of the advancements in the last 5 years that has made this more feasible. We can design guide RNAs to target CRISPR to the gene we're interested in and introduce GFP or RFP tags there. So this will be coming more and more. There is a kind of a problem, but you will be perhaps you will be lacking a functional copy of that protein. It could interfere with the protein function, right? But you should have another wild type protein on the other chromosome, encoded for on the other chromosome. So, but there is that caveat to bear in mind. Now, we've talked about ways to detect certain stages in development where we want to look at what cells exactly are expressing the gene of interest, but there are, of course, high throughput approaches where we can look at expression of the whole genome in the whole animal at different developmental stages. So we can use this microarray and RNA-seq. And of course, this gives a lot of information, right? We can, I'm not sure how familiar you are with these techniques, but basically it's dependent on the RNA expression from the animal that you can then detect via different ways, okay? That's all we need to know for now. But you get the transcriptome of the entire animal at whatever developmental stage you select. But you don't get the specific cell type, for example. You don't know if it's in the CNS or in epithelial tissues or where it's localized in the developing embryo. But you can get a snapshot of every gene that's expressed at that time. And these snapshots usually look like that. So here we have, this is from Flybase. You can go and check that out if you want, where they have done transcriptomics on developing embryos. So here we don't have to go into it, but this is a different stage, and this is a gene. Basically, this is one gene, but there are different probes to the mRNA that can detect its expression over time, and the different time points are here. So you get a lot of information out, definitely, but still, you might find new developmentally important genes that way, right? And then you go and either you make an antibody to the protein, or you do, more likely, you do an in situ hybridization with a probe designed against that sequence to see where in the animal it is expressed, right? So these approaches are complementary. Okay, let's learn some terms that describe the way that cells behave. So we've said before that the potential of individual cells becomes gradually restricted during development. So they become more determined towards a certain fate or a final differentiated state. The number of possible cell types or fates is reduced gradually throughout development. And it has been observed through experimentation, primarily using injection of different colored dyes into different parts of the embryo, that the cells in these separate regions generally follow different developmental programs. So if you inject cells here, they will end up with a clone of cells here, and typically this will be the same set of cells if you repeat the experiment, right? At least it occurs if the embryos that are injected are not subjected to experimental manipulation. So we can surmise that cells within one region proceed to a particular fate, be it that they give rise to part of a limb or an organ or whatever part of the embryo. And then it was, as I said, labeling these cells with dyes that gives rise to what are called fate maps. Then we know that this region of the embryo will give rise to this cell type or be a part of a certain organ or limb. This is a fate map that is already determined when there are no visible structures early on. So, and what they notice that the cells from the same region, they differentiate into the cell type. So if they inject one cell here or one cell here, they will end up with clones of cells that look very much the same. And but these would be distinct to, if you did an injection of cells here, they would give rise to these types of cells here. And by performing these type of dye injection experiments numerous times and observing that when I inject here, it always ends up here, we could make theoretical boundaries in the early embryo or regions that are defined by the positions of these injections, right? So we can build up a map that we know this will always be dorsal and this will always be ventral in the more developed embryo. And scientists wanted to understand this, to study this, and to try and understand how rigid this cell fate was, right? And in order to do this, they did certain kinds of experiments, either explants, where they cut a region of a defined cells they knew they would, the cell fate map, they would develop to, say, a part of a limb or something like that. And they then cultured that part independently from the rest of the egg to see what it would become, to see how rigid was that definition of it, of the cell fate map. Was it for sure it was going to become that part of the more mature embryo or not? And another type of experiment they did was transplant, where they cut from one embryo in one region, and then they placed it into another embryo in a different region to see, how fixed was the fate map. And it's through these kind of experiments that they noted that in some cases the fate was very rigid and sort of already permanent, and in other cases it was more flexible. And they needed terms to describe what they were observing. And this brings us to the term determination, which is a stable change in the internal state of the cell, even if you can't see it in a phenotype. But through these explant and transplant experiments, you could say that they have a stable state. You couldn't change their fate. And this is then most likely caused by permanent changes in gene expression or chromatin modeling. And we haven't discussed chromatin modeling much, but it's adjustments to the structure of the DNA in a way to make the genes be more accessible or less accessible to transcription. So it's again a form of gene regulation. In the case of determination, this change or this stable change or state is inherited by daughter cells. So they remember what they were. There is no external change of the cell's phenotype. The cell differentiation occurs later on. And if a cell is said to be determined, it cannot be changed by external influence, right? It's very stable. It has an irreversible state. And we'll have a look at the experiments they do in the next slide, but I just want to give the definition here. So in contrast to determination, we have specification, whether a cell is specified or not. And it's a small distinction. I say in contrast, but they're similar. So let's see if we get the difference here now. So it is a stable, in inverted commas, change. So not as stable as when a cell is determined, because this internal state of the cell can be influenced by external factors. Okay. Specification is still inherited by the daughter cells, and it occurs then before determination. So it's like a weaker form of determination that is open to influence from external factors. It's only maintained in undisturbed embryos or in a neutral growth medium. So it is not irreversible. We can affect it by disturbing the embryo or positioning the cells in a different place in the embryo, or changing the nature of the growth medium to encourage it to change its fate. So let's have a look at those experiments, and they're detailed in the book. So this was an effort to explain the distinction between specification and determination. So here the fate map shows how the cells would normally develop. We have yellow cells in region A and green cells in region B, And we note that in the differentiated tissue, the cells in the yellow region are hexagon, 6 sides, and they are square in the green tissue in the differentiated state. So what they could do was take a cell from region B in the green and transplant it into the region A yellow cells, and they indicate this transplant by turning the cell red instead of keeping it green. Okay, and what they could see in some cases was that this cell would then differentiate alongside the region A cells. So in this case, it was not determined. We could lift it out and place it into a neighboring region that we know had a different fate map, right? So say this region was going to give us fingers and this region was going to give us toes or something. And what they see is that when they lift out a region that was going to give us fingers and put it into the region that only gives toes, it also gave toes. So it was influenced by the surroundings and therefore it was not determined. In the other case where they performed an experiment in a similar egg, it could be, for example, at a slightly later stage or from a different region. They pick out the cell and they transplant it into the region A here, and they see that as the embryo grows and differentiates, it maintained its differentiated fate even though it was placed into the new region. So it is already determined. It was not influenced by signals in the surrounding area. Even after transplantation, it maintained its cell fate, so it was determined. And then they did the slightly different experiment with explants. So they took out a region here and cultured it in cell media here. So in the absence of signal from surrounding cells, and they saw that it was still able to differentiate into the squares that it should have become. So it was said to be specified. But probably in this case, if you added other cells from the neighbor, you could, for example, encourage these cells to differentiate instead into the hexagon shaped cells. So it's specified, but in that case, not determined. And this is highlighted here in these 2 points, which you can read yourself. I can say that cells are usually determined in early embryos which have a mosaic development. And if you remember, we had 2 types of development. We had mosaic and we had regulated. And in mosaic development, it was where we had these determinants that were passed from mother to daughter cell, and cell interactions were less important. And in the case of regulative development, for example, which is the most common in vertebrates, cells were usually determined later on during gastrulation, for example. And this was shown through these kind of experiments. This slide is a more complicated iteration of the last slide, and it also kind of shows that the state of determination of cells at any stage can be demonstrated by transplantations. And experiments they did here, fate mapping, with regions that normally give rise to the eye. The point of this slide is to show that cells gradually become more determined over time in this system. So if they take from the same cell fate region, early embryo, the gastrula, and they transplant it into a later staged embryo, neurula, they see that the tissue will grow into a normal structure there. It will not grow into an eye. It will grow into a structure appropriate to the animal here, right? So it was not determined. But if we take from the same animal at a later stage, this time we take from the neurula stage, and we transplant it into the same place, into the host animal, we see that in this case, it now develops into an eye-like structure in the wrong place. It is sort of acting, its growth and development is acting independent of the surrounding structures, of the host, the surrounding cells. So it is determined, right? Another way of thinking about this is that this patch of cells at the late stage is insensitive to signals in the from the surrounding host tissue now and is acting autonomously, right, on its own. So cells can can grow and develop in sort of autonomous fashions, or where they are independent of their surroundings, or in non-autonomous fashions where surroundings will play a role. Well, I can hear you ask, that's all well and good, what do these things tell us? What good is this? Can it be used for anything? Well, for a start, I've already described how we can figure out what genes are expressed at what stage, and we can do in situ, to figure out what cells are expressing. So we could, for example, in this case, figure out exactly what genes are expressed here, compare them with what genes are expressed there at this time, right? And see that what are the expression changes over time that lead to a cell becoming determined. And that's one thing, but maybe even more interesting is to look earlier and figure out exactly what's going on at these very early stages. What genes are expressed here that are turned off here, or what genes are not expressed at the early stages that are turned on as a cell becomes more differentiated, right? We could figure that out, and in fact, people have. Uh, they've profiled what the early uh, blastomeres were expressing, and they've used that information to generate stem cells to induce stem cell-like behavior with these induced pluripotent stem cells, or iPS cells, um, that are a type of pluripotent stem cell that can be generated directly from a somatic cell. So a little bit like that in principle where they took the nucleus of a differentiated cell and put it back into the egg. They saw that it held all of the important information, but it didn't have that influence from the egg that was needed to turn on the transcription of certain genes that would give rise to a fully formed adult animal. And we can take differentiated cells from your own body, and now knowing the profile of what they should express when they were young, generate stem cells from them, sort of de-differentiate them. And this was shown in 2006 by the introduction of 4 specific genes. Get these on constructs, introduce the DNA constructs into the cells, express them. Um, and these were, not surprisingly, encoding transcription factors. And the expression of these transcription factors in these differentiated cells was enough to drive them back from differentiation towards a stem cell-like fate. And this, of course, is highly interesting and has been used for in a series of different types of research. So there is a point to knowing, to having this information, and we need the terms to be able to describe it.